You're listening to Health Professional Radio. My name is Wayne Buckler. My guest this morning is Mick Foley, Chief Scientific Officer with research firm AdAlta. Good morning to you, Mick.
Good morning, Wayne.
Mick, first of all, tell us what it is that AdAlta does and what geographic footprint you service.
So, AdAlta is a small biotechnology company based in Melbourne, Australia, and we're working on a novel technology that we use to produce improved antibodies against important diseases such as fibrosis and cancer. And we call these antibodies. And we're really at the early research phase, so we don't exactly sell products, but we are working on the technology and applying it at the moment in animal models and hope to take these through into the clinic.
Now, this is a fairly novel piece of research, I understand. This is not something that's sort of common in practice. Introduce it to our audience. Tell us how you reached this point.
Basically, we're working on this novel technology in a novel area, essentially, which is called next-generation antibodies. And I'm not sure if your listeners are aware, but there's been a fundamental shift in the types of drugs that are being used today. So traditionally, drugs were small chemicals like aspirin and chloroquine and things like that. But now there's a new class of drugs which are biological molecules. These are molecules that are in our bodies, but we've taken them out and we've engineered them in the laboratory. And one of the important components of that is antibodies. It's the things that we all have, the things that are produced when we get infected to fight off infections. And what researchers have done is to take those antibodies and engineer them in the lab so that, for example, there's one called Humira, which is on the market now, which attacks arthritis, and Herceptin, for example, which is for breast cancer. So they're a very successful class of drugs. And in fact, in 2014, five of the top-selling drugs were antibodies. However, they have, even though they are wonderful, they've got some issues that limit their use. They're large, complex molecules, for example. They're expensive to produce, and it's difficult to get antibodies to some important targets like membrane proteins, which means that they're limited in some diseases. And so AdAlta's technology is really a very small, much more stable, very cheap to produce biological molecule, antibody-like molecule that can bind to desired disease targets that are really difficult to attack using traditional antibodies.
And your particular target for these, as you mentioned in the opening, is cancer and fibrosis?
Yeah, so essentially the technology can be applied to pretty much any target because we have a library of these antibodies that we can screen and find different targets. But of course, you have to pick something, and our initial area is fibrosis. And although the antibodies are at the preclinical stage, I think with further research and development and including more extensive animal models and then eventually human safety and efficacy trials, we hope that pretty soon we'll be able to use them in patients with fibrosis. And, I'm not sure, again, if a lot of your listeners be aware that fibrosis is a major disease that affects multiple organs. It's really the formation of excess connective tissue in organs, and it's often thought of as being the healing or repair process gone too far. And one of the areas we're looking at is vitreoretinopathy, which is fibrosis in the eye, in which about 8 to 10% of patients can develop a detached retina, for example. And I guess there's about 5 million people worldwide suffering from pulmonary fibrosis, for example, and that's really got a very, very low survival rate. And although there are currently 2 very recently approved drugs in the market for pulmonary fibrosis, the drugs are actually pretty toxic and they only work in less than 30% of the patients. So there's a real need for alternative treatments in this fibrosis area, which is why we went into it.
Do you have any sense of the antibody toxicity response? Is it because it comes from antibodies? Does it have a much lower toxicity?
Yeah, well, this is right. It's because it's a biological molecule and because antibodies are exquisitely specific. It means that they've got very less off-target effects, like, for example, some of the common chemical drugs. Those drugs have often, Vioxx, the story of Vioxx, where it was a wonderful drug for arthritis, but it caused a lot of issues. there's much less chance of that happening if the experiments are done carefully and you target it, because those antibodies tend to be absolutely specific.
Mick, it sounds like it's fascinating research. When you say your timeline was shortly they'll be able to be used, shortly is one of those words in medical research. How long are we actually talking?
Yeah, so look, I mean, there's a joke in this research that it's 5 years away and it always will be. I think really, what we've done very recently is show that our antibodies, when we inject them into mice, will block the immune cells in those mice from migrating towards an inflammatory response. So we were pretty excited about this because it's the first time that we've really shown that these molecules in an animal will actually do something useful. And so it's obviously a major step forward. But really what we need to show is that there is a real effect in a couple of models of animal fibrosis. And then we're looking at putting them into human trials. Now, we're talking to people already about doing a first-in-human trial. But really that is probably, and again, it all comes down to resources, but realistically you're looking at, 18 months to 2 years, if everything goes well, where we could actually put these into humans.
Mm-hmm. Now, forgive my gross ignorance of medical research here. How do you go about making a molecule?
There's many ways. I mean, we use genetic engineering, which is a really common technique in pretty much every molecular biology lab in the country, in the world, in fact. And really what we do is we have a library of these tiny molecules, and we've got them expressed on a bacterial virus, and that means that we can grow them continuously. We never have to go back and try and remake them. They're pretty much there, and they're immortalized. And then when somebody comes to us, in fact, we've done some contracts for companies like Roche who have come to us and said, look, we would like one of your antibodies against a particular molecule that's involved in inflammation or autoimmunity. We will then go to the freezer, take our library out. We'll then screen that library. We call it panning because you've got many millions and billions of these molecules, and only one or two of which will be actually useful in binding. And so you sort of pan like panning for gold. You try and get rid of all the rubbish, and you end up with one or two binders. Now, even then, you've still got to then improve those, make them better. So you do a bit of mutation. So it's really taken AdAlta many years to actually come to the stage where we can even begin to put these into animals. Now, all that research is done sort of under the radar, if you like, before we even go out and talk to people.
Mick, it's fascinating to talk to you. It's really the very leading edge of the stories that we normally do. Generally, when we're talking with pharmaceutical companies, it's because they've got a new product on the market, and talking with you is talking 5 or 10 years before that happens. But it's fascinating to hear the story, and it's great to hear that it's happening in Australia.
Terrific. Yes, well, it's been great to talk to you. I hope your listeners get something out of this.
Well, I'm sure they will. I'd like to leave you with one last question. What's the greatest misconception about your work that drives you nuts and keeps you awake at night?
Yeah, look, I don't know if there's anything that really drives me nuts, except when my favourite soccer team, Glasgow Celtic, get beaten. But I guess what I didn't mention before is that these antibodies are actually human molecules. And that means, like you touched on before, they should have very few issues with toxicity or, for example, being rejected. But what AdAlta actually started working on very similar molecules from sharks.
All right.
So sharks are actually one of only 2 animal species that produce these small, stable antibody-like molecules. And we've got a lot of data suggesting that they're really useful. However, we took the information from the shark molecule and designed essentially a human version of that. And that's great for us because, as I said, there should be no adverse reactions. But I guess the problem for us is that AdAlta, many people think of AdAlta still as a shark company. And I think that's probably because people like hearing about sharks, and the story is actually quite nice, especially, from an Australian company, as we've got a lot of sharks in Australia. But it is a bit frustrating that people haven't really caught up with the fact that we've learned everything we've needed from the shark, from the evolution of the shark, and we've moved on to create a technology which has got all the positives of the shark molecule, but because it's human, it should have a much safer use in therapy.
Well, with a little bit of luck, there'll be a few more people in the industry today who don't think of you as such a shark company. For people who want to know about AdAlta, the website is AdAlta, adalta.com.au, and we've been talking with Mick Foley, their Chief Scientific Officer. This is Health Professional Radio. A transcript of this interview is on our website, along with a sound archive at www.hpr.fm.